And the objectives for the first part of the lesson are to define the two divisions of the nervous system, list three general functions of the nervous system, discuss the cellular composition of the nervous system, including compare the structure and functions of the neuroglia and neuron, explain the function of the myelin sheath, explain how a neuron transmits information, describe the structure and function of a synapse. So for divisions of the nervous system, the nervous system coordinates and directs the various organ systems of the body. The central nervous system includes two structures, which are the brain and the spinal cord. The peripheral nervous system consists of the nerves that connect the central nervous system with the rest of the body. It is located outside the CNS. The sensory nerves gather information and carry it to the central nervous system. The integrative function serves to process or interpret the information brought to the CNS by sensory nerves. The motor nerves convey information from the CNS to the muscles and glands to carry out the plans made by the CNS. So we have the old example of putting your finger, touching a hot stove, and that information is sent from the sensory nerves to the CNS indicating it's hot, and then a motor impulse is sent back for you to pull your fingers away, and that happens, of course, instantaneously. But that would be an example of the interpretation of the sensory information and being sent back to the motor nerves. Okay, now for cells that make up the nervous system, there are neuroglia or glial cells. These are the most abundant type, and they support, protect, insulate, nourish, and generally care for the neurons. Two of the more common glial cells are the astrocytes and the ependymal cells. The star-shaped astrocytes are the most abundant of the glial cells and have the most diverse functions. Neurons do the communicating for the nervous system, and their long shape makes them delicate. Unlike glial cells, neurons are non-mitotic and therefore do not replicate nor replace themselves when injured. Neuroglia. Only two of the 5 types of glial cells are shown on this particular slide. The astrocytes form what's called the blood-brain barrier, and we'll talk about that as part of the blood-brain barrier topic. The ependymal cells help form cerebrospinal fluid, or CSF. If you take a look at Table 10.1, you'll be able to see the functions of each type of neuroglia, and that's located on page 175 of your text. Next, we see a neuron, and the neuron is the second type of nerve cell, and it's the most abundant in the transmission of information. So we see the parts of the neuron on the slide: the cell body with the nucleus, the dendrites which branch off of the cell body. The long structure we see there is referred to as the axon, and then the structures that branch off of the axon are called axon terminals. The electrical signal travels from the cell body down toward the axon terminal, as this arrow illustrates here. This slide also illustrates some parts of a neuron. You see the axon, which we saw on the previous slide. This shows you what's called the myelin sheath, which surrounds the axon. The nodes of Ranvier, and you can see those little areas that go in that are pointed out in this illustration, are not covered by myelin. They are considered unmyelinated. There's also a structure that separates myelin called neurolemma, and this is often found in the peripheral nervous system, but also in some unmyelinated areas of the CNS. And then you see the axon terminal. So the myelin insulates and protects the axon. And the formation of myelin differs in the peripheral nervous system and the central nervous system. Now for types of neurons. The sensory neurons or afferent neurons carry information from the periphery toward the central nervous system. The motor neurons are called efferent neurons. They carry information from the central nervous system toward the periphery. Both sensory neurons and motor neurons are found in the central nervous system and in the peripheral nervous system. Interneurons are found only in the central nervous system. They form connections between sensory and motor neurons. So we find both white matter and gray matter in the central nervous system. So what's the difference? The white matter is made up of myelinated axons. The gray matter is made up of unmyelinated axons, cell bodies, interneurons, and synapses. So clusters of cell bodies located in the central nervous system are generally referred to as nuclei. Small clusters of cell bodies in the peripheral nervous system are called ganglia. So what is the nerve impulse? Electrical signals convey information along a neuron. It's also called the action potential, and the signal moves along sensory or motor neurons. A series of events cause the electrical charge inside the cell to move from its negative resting state to its positive depolarized state and back to its negative resting state again. And we'll look at that in the next slide. Polarization is the resting state of the nerve impulse, and the inside of the neuron is more negative than the outside when it is polarized. Depolarization is the stimulated state, and this is the change inside the cell from negative to positive when the neuron is stimulated. And then there's repolarization, which is the return to the resting state, and the inside of the cell becomes negative again. What causes the nerve impulse? The movement of specific ions across the cell of the neuron is responsible for the action potential or nerve impulse. During the resting state, the potassium ions leak out of the cell, taking the positive charge with them. We know that potassium carries a positive charge. This leakage is responsible for the resting membrane potential. The inward diffusion of sodium ions causes depolarization. And then the outward diffusion of potassium causes repolarization, returning the cell to a resting state again. So you have polarization with the potassium leaking from the neuron. It determines the resting membrane potential. Then repolarization where the sodium rushes in. Depolarization where the sodium rushes in. And then finally repolarization where the potassium rushes out. What causes the nerve impulse to move? The terms nerve impulse and action potential mean the same thing. A nerve impulse or action potential must move the length of the neuron. So if you refer to figure 10.7 on page 179 of your text, each nerve impulse or action potential has the ability to depolarize the adjacent membrane. This causes the nerve impulse to move toward the axon like a wave. The height of each nerve impulse is the same along the entire length of the axon. This ensures that the nerve impulse does not weaken as it travels the length of a long axon. The action potential is responsible for the release of neurotransmitters. So we're aware that the nerve impulse or axon potential moves very quickly. So how does that happen? It mentions here that the myelin insulates the axon, and the myelin exposes some axonal membranes, and we see that at the nodes of Ranvier, those demyelinated or unmyelinated, areas of the axon. And then the action potentials jump quickly from node to node like a kangaroo. So the action potential jumps from this node to the axon terminal. The jumping kangaroo effect increases the speed of the action potential. The jumping of the action potential is also called saltatory conduction. Myelinated fibers are therefore very fast conducting fibers. So let's start looking at the synapse. The synaptic cleft, there are receptors, neurotransmitters, and inactivators in that synaptic cleft. And what are receptor sites? They're places on the membrane to which the neurotransmitters bind. The most common neurotransmitters are acetylcholine and norepinephrine. Inactivators are substances that terminate the activity of the neurotransmitters when they have completed their task. So what are the events at a synapse? If you would refer to figure 10.9 on page 181 of your text, you will see that there is an illustration of how the nerves communicate and the various steps. So acetylcholine, which is a neurotransmitter, is secreted from neuron A. It diffuses across the synaptic cleft. It binds to the receptors on neuron B, and neuron B is then activated. So nerves must communicate with one another, and they do so at the synapse. In the upper panel, the boxed area shows the communication at the synapse, and then the lower panel shows the steps. The remaining objectives for the next part of the lesson are: describe the functions of the four major areas of the brain and the four lobes of the cerebrum. Describe how the skull, meninges, cerebrospinal fluid, and blood-brain barrier protect the central nervous system. We know that the brain is located in the cranial cavity, and it's pinkish gray in color. It's delicate and has a soft consistency. And it's divided into four major areas, which you see here illustrated on the slide. The primary source of energy for the brain is glucose. So you see the large area is the cerebrum. The diencephalon, you see, is pointed out. We'll talk more about it later. The brain stem and the cerebellum. So an earlier slide referred to gray matter and white matter, and the gray matter of the cerebral cortex allows us to perform higher order mental tasks, such as learning, reasoning, language, and memory. The white matter is composed primarily of myelinated axons that form connections between the parts of the brain and spinal cord. Markings that we'll find on the cerebrum. The gyri, the singular is gyrus, or convolutions, look like speed bumps on the surface of the brain. By increasing surface area, they increase the amount of tissue available for thinking. Fissures, or sulci, are grooves that separate the gyri. Of particular interest is the central sulcus, which separates the frontal lobe from the parietal lobe. It's an important landmark on the cerebrum. This slide illustrates the lobes of the brain, and it is figure 1011 in your text. I believe it's on page 183, 184. So figure A shows the 4 lobes of the cerebrum: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. If you look at figure 1011, in what central lobe is the precentral gyrus found? The precentral gyrus is found in the frontal lobe. In what cerebral lobe is the postcentral gyrus found? The postcentral gyrus is found in the parietal lobe. If you refer to page 184 in your text, there's a table, 10.2, which is a listing of brain structures and their functions. The frontal lobe may be referred to as the executive and is responsible for behavior, personality, and motor control. So most frontal lobe functions are associated with higher order intellectual functioning and motor activity. So if a patient or a person sustained a severe injury to the frontal lobe, they might have difficulty with memory, changes in their personality and behavior, and impairment of intellectual and motor functioning. They might even need to relearn how to read. So as far as motor activity, the frontal lobe contains the precentral gyrus, which is the primary motor area, also the frontal eye field, which controls voluntary movements of the eyes and the eyelids, and the motor speech area called Broca's area. So if a person were to suffer a stroke that involved Broca's area, they would be able to understand what you were saying, but might not be able to respond verbally. And the reason being is Broca's area deals with the physical aspects of producing speech, not the cognitive issues such as understanding the meaning of words. This slide shows all the functional areas of the cerebrum, and I would suggest that you look at page 183 of your text and review these. In this slide, the homunculus represents the amount of brain tissue that corresponds to a motor function of a particular body part. Each part of the body is controlled by a specific area of the primary motor cortex of the precentral gyrus. Complicated movements require large amounts of brain tissue. So take a look at the homunculus's hands and feet. The hands are much bigger than the feet, and the reason being is that the hand performs more complex movements that require more brain tissue. Looking at other cerebral lobes, we have the parietal, the temporal, and the occipital. The parietal lobe contains the somatosensory area. This area interprets temperature, pain, pressure, light touch, and proprioception. And proprioception is the way we're able to feel our body in space. The temporal lobe contains the auditory cortex and the olfactory area. Information from the taste buds is interpreted in both the temporal and parietal lobes. The occipital lobe contains the visual cortex. Speech areas span the temporal, parietal, and occipital lobes, usually in the left hemisphere of the brain. The association areas help to interpret sensory information. Examples would be visual, auditory, and somatosensory. Injury to the left hemisphere is much more likely to result in speech impairment than injury to the right hemisphere, and the reason being that speech centers are concentrated in the left hemisphere. The primary visual cortex allows us to see an image, for example, a tree. So what additional information does the visual association area provide? The visual association area allows you to interpret or apply meaning to the image, perhaps that it is an oak tree or that it needs water. What are basal nuclei and what do they do? Scattered throughout the cerebral white matter are patches of gray matter called basal nuclei. Sometimes they're referred to as basal ganglia. The basal nuclei help regulate body movement and facial expression. Dopamine, which is a neurotransmitter, is largely responsible for the activity of the basal nuclei. There are two parts of the diencephalon, the thalamus and the hypothalamus. The thalamus acts as a sensory pathway for information coming from the lower brain and the spinal cord to the sensory areas of the cerebrum. It is especially important with regard to pain sensation. The hypothalamus sits above the pituitary gland and controls endocrine function. It is also the body's thermostat and helps regulate autonomic functions. So we talked about the hypothalamus in the integumentary system and how its role was the thermostat of the body. The midbrain contains nuclei that serve as reflex centers for vision and hearing. The pons plays an important role in regulating breathing rate and rhythm. The medulla oblongata descends through the foramen magnum as the spinal cord, and you should remember the foramen magnum from the study of bones. It is called the vital center because it controls heart rate, blood pressure, and respiration. Its emetic center stimulates vomiting, either as a result of direct stimulation, for example, fear, spinning, or distressing odors, or from indirect stimulation from the chemoreceptor trigger zone, or CTZ, as in cancer chemotherapy. So if you remember from medical terminology, emesis means vomit, and emetic would be pertaining to vomiting. The cerebellum is connected to the brainstem by 3 pair of cerebellar peduncles. These stalks or connections allow the cerebellum to receive, integrate, and deliver information to many parts of the brain and spinal cord. The primary responsibility of the cerebellum is the coordination of voluntary muscle activity. It produces a smooth, coordinated muscle response after integrating all the incoming information from many areas throughout the body. It is also a major reflex center. Now we will address structures across divisions of the brain. These three important structures are not confined to any one division of the brain. They overlap several areas. The limbic system is a wishbone-shaped group formed by parts of the cerebrum and diencephalon. The reticular formation is responsible for maintaining consciousness and activating the sleep-wake cycle. It also contains the gaze center. Consciousness is a state of wakefulness and is dependent on the reticular activating system. So the levels of consciousness include attentiveness, alertness, relaxation, and inattentiveness. The exact cause of sleep, according to our text, is still unknown. Coma is a hypo-responsive state with several stages ranging from light to deep coma. Let's talk about the stages of sleep. Although we may not know the exact cause of sleep, we know that it's essential to our well-being. And here we'll address non-REM or rapid eye movement sleep and REM or rapid eye movement sleep. The four stages of non-rapid eye movement sleep progress from light to deep. Rapid eye movement sleep is characterized by fluctuating blood pressure, respiratory rate and rhythm, and pulse rate. The most obvious characteristic is rapid eye movements, for which the sleep segment is named. Areas of memory include immediate memory, short-term memory, and long-term memory. The memory areas allow the recollection of thoughts and images. Many areas of the brain are responsible for memory. There are four layers involved in protecting the central nervous system. They are bone, the meninges, cerebrospinal fluid, and the blood-brain barrier. The bone would consist of the cranium and the vertebral column, which would help as protection for the central nervous system. We'll address the meninges, the CSF, and the blood-brain barrier in upcoming slides. So first of all, the meninges. The meninges are membranous layers that surround the brain and the spinal cord. And the pia mater is the innermost layer. It is a very thin membrane and it contains many blood vessels. The arachnoid layer is a spider web-like membrane between the pia mater and the dura mater. The subarachnoid space surrounds the entire central nervous system and is filled with cerebrospinal fluid, or CSF, to help cushion the central nervous system. The dura mater is the outermost layer. It's a thick, tough, connective tissue that splits into sinuses filled with blood. The subdural space is located below the dura mater. It contains blood. If a person sustains a sharp blow to the head and develops a subdural hematoma, what symptoms might they have? What happens is because the cranium cannot expand, a subdural hematoma causes pressure on the brain. The symptoms depend on the location of the pressure, but might include a dragging foot, blurred vision, slurred speech, or a decreased level of consciousness. Cerebrospinal fluid. The cerebrospinal fluid, or CSF, is composed of water, glucose, protein, and several ions, especially sodium and chloride. If you refer to figure 1015 on page 192 of your text, you'll be able to see the two lateral ventricles, the 1/3 ventricle, and the 1/4 ventricle, and the cerebral aqueduct. The CSF is formed within the ventricles of the brain by the choroid plexus, or the ependymal cells. CSF is a clear fluid and is similar in consistency to plasma. Some of the CSF flows through the central canal, which is a hole in the center of the spinal cord. It eventually drains into the subarachnoid space at the base of the spinal cord. A second drainage path is through the foramina near the brain. Drainage of cerebrospinal fluid. The amount of CSF drained must equal the amount produced. What if the canal between the 3rd and 4th ventricles becomes blocked, perhaps by a congenital malformation? The CSF would then accumulate in the ventricles, increasing intracranial pressure and pressing on brain tissue. Treatment could include shunting the CSF around the blockage. Our last slide addresses the blood-brain barrier. Glial astrocytes are part of the cerebral blood vessel wall that supplies the brain and spinal cord. These astrocytes select the substances allowed to enter the CNS from the blood. Some antibiotics or drugs in general cannot cross the blood-brain barrier. In the case of an infection, how could the CNS be treated? An infection could either be treated by selecting an antibiotic that does cross the blood-brain barrier or injecting the antibiotic into the subarachnoid space. Please be sure to carefully review your material, do the end of chapter review, and I hope you enjoy the study of the central nervous system.